Dear students, hope you have enjoyed summer vacation, and we had a very long summer vacation because of the pandemic COVID. So anyhow, we have to start our third semester. So in the last two semesters, we covered the basic principles of biochemistry, which includes the different techniques which is used and also different types of biomolecules. So in the third semester, we have to cover the metabolic aspects of biochemistry. So you know something about metabolism. Metabolism is the chemical reactions which is happening in a cell, which leads to generation of many molecules, utilization of many molecules. So metabolism is comprised of anabolism and catabolism, which we will study later. So when you talk about the third semester syllabus, we have 4 units, of which the first unit is enzymes. So the enzyme unit covers almost every aspects of enzymes, including the chemical nature of the enzyme, their functions, their applications, and how these enzymes are classified in a scientific basis, and how these actions of enzymes are affected by different physical factors and the chemical factors also. In addition to this, we have to study the kinetics. So kinetics is all about how the different conditions affects the velocity of an enzyme catalyzed reactions. So it's a little bit difficult portion, which comes a little bit of graphs and plots and other things. Next, we have to cover the specificity of the enzymes. So we know enzymes are specific, but the degree of specificity varies from one enzyme to other. So we have to also cover that portion also. Next is how these enzymes are inhibited. So there are different types of inhibitions, and there are different types of molecules which inhibits the chemical reactions, that is the enzyme modulated or enzyme based reactions. So there are not the inhibitors alone. Some molecules will act as activators. So we have to study how all these will influence the chemical reactions. Next, of course, the applications of the enzymes. So enzymes are applied in various fields like industry, medical, that is the clinical aspects, bioremediation and food industry, and it is almost applied in every fields. So it's very important to learn the basis of enzymes. So that is the only thing which we have to cover in the enzymes, which is a little bit a very huge topic. So after studying the enzymes and its characteristics, so we have to move to the metabolic aspects. So initially, the metabolism is divided into two portions. That is the first portion, that is the carbohydrate metabolism, is dealt in the third semester, and the rest of the metabolism will be covered in the fourth semester. So the second unit is all about anaerobic metabolism of carbohydrates. So which includes the basic metabolic pathway, which details how the carbohydrates are being digested, absorbed, and after absorption what will happen to different carbohydrates. Especially we focus on the glucose, because glucose is the main energy source of every cell, whether it is plant or animal. So one of the things which we have to remember is that our syllabus is mainly based on animal metabolism rather than the plant metabolism. So we just give some stress to animal metabolism. And of course, we have one unit which covers the photosynthesis. So in the second unit, which is the anaerobic metabolism of carbohydrates, which depicts that these metabolic process doesn't require any oxygen. So these metabolic steps will happen in absence and in presence of the oxygen. So it's an anaerobic metabolic process. So which includes glycolytic pathway. So we know we have studied or we have come across glycolytic pathway in earlier classes. So you might be very familiar with glycolysis, which is very important metabolic pathway for every cell. So so that is all about the glycolysis. Next is fate of pyruvate. The end product of the glycolytic pathway is pyruvate. So we have to see what happens to this pyruvate and what are the fates of what are the fates of pyruvate. The pyruvate has many fate depending on the condition or the metabolic state of a cell. So after studying all these, we have to go to glycogenesis and glycogenolysis. So what is glycogenesis and glycogenolysis? So we know if we take excess of glucose, our body will store the excess of carbohydrate, which is the glucose, and the excess of glucose will be stored in glycogen, that is mainly in the liver. So that process, the formation or the conversion of glucose to glycogen is glycogenesis. And glycogenolysis is whenever our body is lacking a little bit of glucose, the body has to maintain that glucose level, mainly in the blood. So what our body do is that it takes the reserve glycogen, that is the glucose which is stored in the liver, and it has been channelized or degraded to glucose, and the glucose is flushed into our blood, thus maintaining the glucose level. So we have to study what are all the different steps, what are all the enzymes and coenzymes and other things which are involved in these two processes. The other portions which is in this unit is the role of cyclic AMP, that is CAMP. We know the hormonal activity. So for hormonal activity, cyclic AMP is a very important molecule. So we have to study the roles of cyclic AMP in glycogen metabolism. Next is gluconeogenesis. So what is gluconeogenesis? Gluconeogenesis is a metabolic pathway in which glucose is synthesized from non-carbohydrate sources like amino acids, pyruvate, and other things. So production of glucose from non-carbohydrate sources is called as gluconeogenesis. So we have to study the metabolic pathway and the enzymes in it. Next is pentose phosphate pathway, which is otherwise called as HMP shunt, that is hexose monophosphate shunt. So what is the purpose of pentose phosphate pathway? This produces very important molecule like pentoses, especially riboses, which is utilized for the synthesis of RNA and DNA. We know DNA and RNA contains deoxyribose sugar and ribose sugar respectively. So these are provided by pentose phosphate pathway. Not only this pathway provides ribose, but also it supplies reduced NADP, that is the reduced form of a coenzyme which is called as NADPH plus H plus. And this reduced NADP is used mainly for the fatty acid synthesis. We know our body needs fats, so the fat has to be synthesized. So for those synthetic purposes, fatty acid synthetic purposes, we use this reduced NADP. So that is all the basic anaerobic metabolic pathway which we have to cover. And remember, we don't have to study the structures. So if you are interested in, just let me know whether I should take it in the structure wise explanation, or for the time being it is not mandatory. So anyway, it is always better to study the things which with structures, because it helps you in the post graduation. So next unit is aerobic oxidation of carbohydrates, which is the third unit, under which we have to study decarboxylation of pyruvate. So the pyruvate which is thus produced in the glycolysis will be channelized or will be utilized. That means during glycolysis, we take some energy of the glucose, which is resided in the glucose, into ATP. So rest of the energy is with the pyruvate. So we have to tap that energy also. For tapping or getting that energy from the pyruvate, which is formed from the gly glycolysis, has to be utilized or has to be tapped. For this reason, the decarboxylation of pyruvate happens, and which channelize or which puts this pyruvate into very famous citric acid cycle, which you have already covered to some extent. So citric acid cycle is the cycle in which the pyruvate, even though it is not directly entering, the pyruvate is converted to acetyl-CoA, and then it will enter into the citric acid cycle. So once the citric acid cycle is happening, this leads to the complete oxidation of the carbon molecules in the pyruvate. So the carbon in the pyruvate is released as carbon dioxide. So by the time the pyruvate gets itself oxidized and the energy is released and the energy is tapped at various reactions of the citric acid cycle, and the energy is trapped in ATP molecules. So for this to happen, oxygen is necessary. Suppose there is no oxygen in a cell, the citric acid cannot undergo, because it ultimately needs oxygen, even though it doesn't need direct oxygen. So why we call citric acid is a aerobic process? Because after the citric acid cycle, the equivalence which is produced, that is the ATP, and the reduced components, this has to be oxidized. And for oxidation, these reduced components like FADH2, NADH2, these are all nothing but reduced coenzymes. So these coenzymes should enter into electron transport chain, which is otherwise called as ETC. In ETC, the finer acceptor molecule is oxygen. So ETC will happen, or it will be performed only if the oxygen is present. So if the oxygen is absent, ETC cannot happen. So because of the sluggishness in the ETC, the citric acid cycle, the products of the citric acid cycle accumulates in the cell, which leads to the inhibition of the citric acid cycle. So that is why we say citric acid is a aerobic process. So next, in the later part of this, the third unit, you have to study the mitochondria and its basic arrangement. We know the reduced equivalence, as I have told earlier, the TCA cycle will produce this reduced equivalence like NAD and FAD, H2. So these reduced equivalence has to be channelized to electron transport. And what are electron transport? What is electron transport? These are all nothing but a series of proteins which are arranged in the increasing order of the redox potential. You know what is redox potential is? That is the capacity of a molecule to reduce or oxidize the other components. So in ATC, you have a series of proteins which is arranged in the increasing order of their oxidation reduction capacities. And these electron, that is the components of the electron carriers, is present in the inner mitochondrial membrane. You have studied the mitochondrial structure. So inside the inner mitochondrial membrane, you can see these components, that is the components of the electron carriers. So we have to study the cytology of the mitochondria and how these reduced components are arranged and things like that. So after understanding how these are arranged, we have to see how these reduced coenzymes like FADH2 and NADH plus H plus will enter into the ETC and how it gets oxidized. So during oxidation, the electrons is transported from these reduced equivalents to these components, that is the electron ETC components, and finally the electron is asserted by oxygen. So without this oxygen, that is the final carrier of the electrons from reduced equivalence, ETC cannot perform. So it is very important that the cell should have sufficient oxygen to take up the electrons from the reduced equivalence. So that is why we respire. Basically, we respire. We take the oxygen, that is the air, which contains a good amount of oxygen, and it is distributed into the blood, and the blood supplies the oxygen to every cells so that the oxygen potential is being maintained, which sufficiently provides the oxygen for the electron to be charged into the oxygen. So we have to study it in detail, and during this electron transfer, energy is released, and this energy is trapped in ATP, which involves a special ATP synthase complex, which is also in the inner mitochondrial membrane. So there are different theories regarding how the energy released from ETC is coupled to ATP synthesis, or we call it as the phosphorylation. That is, what do you mean by phosphorylation? Phosphorylation is the production of ATP. So the energy which is released from the ETC is utilized for the ATP production. So there should be coupling, that is, the energy has to be transferred to phosphorylation. So how it is happening? So there are different theories, and finally we know that it is the chemiosmotic mechanism. So we have to study what is the mechanism and how these being coupled. So it is a little bit complicated thing. The other topic which we have to cover is what are all the high energy compounds. That means we know some molecules have some residual energy within the molecule itself, and the degree of energy which is resided in each molecule is a variable one. Some have very high energy, some have very low energy, and some have intermediate free energy. So our body needs energy basically for different things like motor actions, that is the movement, growth, growth, that is the division of the cell, then other metabolic process. So it requires energy. So for these energy to be obtained, we require high energy compounds like basically ATP. So we call it as the energy currency of the cell. So we need energy currencies. So like that ATP, there are some other high energy molecules like creatine phosphate and some other molecules like that. So these are the high energy compounds. So we have to study what are the different types of high energy compound and how you can call a molecule a high energy compound. So like that. Then we know the electron transport chain works by chemiosmotic theory. So there are some chemicals which inhibits ETC. Some are acting as uncouplers, some are acting as inhibitors. So there are many. Most of the toxin or some of the toxins are targeted to ETC. One of the famous toxin is cyanide. You know, cyanide is very toxic, very toxic. So what the cyanide do is that if you take cyanide, it blocks the transfer of electron from ETC to the final acceptor molecule, which is oxygen. So ETC cannot happen. Thus, TCA cannot happen. So what happens? The cell doesn't gets the sufficient energy, that is the ATP, and the cell cannot perform, and ultimately cell dies. So that is what is happening when you take cyanogen. So cyanogen toxicity is like that. So we have to study uncouplers. What are uncouplers? What are the different types of uncouplers and examples, and how do they deliver their function, or how do they affect on etc. And there are inhibitors also. We take different types of antibiotics. So what they do is that they go and interfere with the etc of the pathogen, so that the pathogen cannot survive and it ultimately dies. So there are many compounds which is targeted to etc. So this is about the aerobic respiration. So we covered aerobic as well as the anaerobic. So the fourth unit is photosynthesis. So you know what the photosynthesis is all about. What are the different reactions which is involved in the photosynthesis? As we all know, photosynthesis is the primary or the very important metabolic pathway which keeps the life in earth. Without photosynthesis we could not, because especially we are animals, we are all consumers. So the photosynthesis mainly happens in producers, that is mainly in plants and some algae. So we have to cover the reactions, different types of reactions. Basically, photosynthesis includes light dependent and light independent, or we call it as the light reaction or the dark reaction. So we have to study what are all the pigments involved in the photosynthesis. We know different pigments, leaves and plants, that is the plants contains different pigments. Each individual pigment will absorb light at a particular wavelength. So we have to see what are all the different pigments, what are all the wavelength they absorb. So finally, when the light is being absorbed by this, it causes light reactions. Light reactions, that is production of ATP, oxygen, and other components. So in the next stage is we have the dark reaction, that is the fixing of carbon dioxide. That means the air contains carbon dioxide, and the carbon dioxide is fixed during this dark reaction with the use of compounds like ATP and other things which are produced in the dark reaction. So the producers fix the carbon dioxide through dark reactions, which is otherwise called as the Calvin cycle. So by the Calvin cycle, the carbon dioxide is fixed and we will get the carbohydrate, basically glucose, which is the important source of energy. So we have to see what are all the steps involved and the enzymes involved. And we have to also see the C4 cycle, which is an alternative pathway, and we have to study glyoxylate cycle, which is a cycle which involves some initial reactions of the TCA cycle, which happens in some groups of animals. We don't have any glycosylate cycle. So we have to study its significance also. So these are the 4 units which we have to cover in this semester. So for referring these topics, you can either go Satyanarayana, which is the very basic book, or you can go either to Wyatt, Leninger, Matthews. There are many. So it is up to you to whether which book you have to refer. So these is about our third semester syllabus. So one thing I would like to say is that we have some portions which is left in the second semester, basically some equations like ionization of water, Donan's equilibrium, things like that. You might have studied in the +2 or in the previous class. If you have any doubt regarding any of the topic of the second semester, please inform me. I will arrange an online class for it. So these are all the things. So next we can see the enzymes. So we can cover the syllabus according to the syllabus itself. We don't have to jump to last units, which I have done it in the second semester. So it's a very well arranged syllabus. So we can start from enzymes. So in the next class, we will see, or we can start the enzymes. So enzyme is a little bit difficult. One of the important thing which I have to say while studying the enzyme is that you have to understand each and every point of this unit. So unless you skip, if you skip some points, it makes some confusions in the later stages when you come with kinetics, or when we deal with kinetics, inhibition, and all, you will be little bit confused. So try to understand every point as far as possible, and don't be reluctant to ask questions. So you can ask questions either through WhatsApp, or you can call me. You have many options. So it is always better to call me after 8 p.m. so that I will be free, and you will also be free. There is no stringent time stipulations. You can call me at any point. So that's all. I hope you got a overall picture of the syllabus of the third semester. So kindly attend the videos and learn as far as possible. and kindly come to the online classes. That is, actually it is not an online class, it is just a discussions. So for an effective discussions, you have to just go through the class which is uploaded, and let us have a discussion of about half an hour or more. It all depends. Don't hesitate to ask questions during the class. So okay, so nothing more. Take care. Thank you.